In this video, we are going to talk about antigen presentation. The presentation of antigens by all of the cells in our body actually help to connect the innate immune response with the adaptive immune response. Particularly, we will focus here on the role of macrophages and dendritic cells in presenting antigens within the lymph nodes to T cells. Before we get into the details of antigen presentation, I think it's best that we take a couple of minutes to review the innate immune response, particularly focusing on inflammation and phagocytosis. The image on the right here, which comes from OpenStack's microbiology, you'll remember, is a representative image of the process of inflammation. In this process, neutrophils, macrophages, and dendritic cells are recruited to the site of infection by the secretion of cytokines and chemokines from the cells that are at this site of the infection already. We know that neutrophils and macrophages are phagocytes, and they're going to then perform phagocytosis and remove waste by exocytosis. However, some of the macrophages that are located here are actually going to travel through the lymph system to lymph nodes where they will interact with T cells and present antigens to those T cells. So what is an antigen? Well, an antigen is anything that stimulates an immune response. Particularly, the name antigen comes from antibody generating. Particularly, antigens are molecules that are going to stimulate the production of antibodies. Antigens, as shown in this picture, are a whole molecule, like a whole protein, whereas epitopes are subsets or parts of the antigen. And so, for example, in here, we're looking at three different shapes, something that looks like a pyramid, something that looks like a sphere, and one that looks a bit like a box. And these three epitopes here would be parts of a protein, maybe about 20 or so amino acids in size, but each of them are a part of an antigen. Pathogens, then, which are organisms that cause disease, are made up of multiple antigens. Now, to review, the lymph system is a system of vessels as well as organized tissue where, and in some cases organs, where we find white blood cells and other cells that are going to be involved in immune response. So how does a cell travel to these lymph nodes? Well, for example, if you have a cut in your hand, what you would find if we follow this kind of red line here up towards the armpit, macrophages would enter into lymph vessels and travel to the location of lymph nodes. And these circles here highlight some of the major locations of lymph nodes within our body. These are not all of them, but areas include under the armpits, within the neck, and also within the groin. We might also find that cells will localize to the spleen in the case of white blood cells and B cells in particular. But it's at these locations where these innate immune cells, particularly macrophages and dendritic cells, are going to begin to stimulate the adaptive immune response. And that's going to be the focus of our topic here. So let's briefly talk about this process of presentation before we get into the details of how it's performed. In this broad overview, what we're going to focus on are the receptors that are involved in this presentation. These receptors are known as major histocompatibility complexes, or MHCs. And there are two different types of MHC, MHC1 and MHC2. On these receptors, they are found either on all cells, in the case of MHC1, and I should preface this by saying all cells with a nucleus. And then MHC2 is found on what we call professional antigen presenting cells, or PAPCs. This includes macrophages, dendritic cells, and B cells. And finally, these two also differ in what they are going to present. MHC1 oftentimes is associated with presenting intracellular epitopes, or particularly antigens from intracellular pathogens like a virus or a parasitic bacterium that needs a host cell in order to replicate. On the other hand, MHC2 will present epitopes from pathogens that have been phagocytized, so those that have been taken up from the outer environment, engulfed by the cell, and now presented rather than using the debris as waste. These professional antigen presenting cells are going to use that debris at the end of phagocytosis for antigen presentation. Let's now take a look at the details of MHC2 and its presentation. Now, MHC2 will be found on what we call professional antigen presenting cells, and I've already defined that these professional antigen presenting cells include macrophages, dendritic cells, those are both part of the innate immune response, and then also B cells are considered to be professional antigen presenting cells. One of the things that these cells all have in common is their ability not only to perform phagocytosis, but then to present parts of a pathogen on their surface on MHC2. Parts of this pathway here are going to look very similar to what we've talked about in the past, as it does involve the process of phagocytosis. Let's assume that we're working with a dendritic cell here, and you notice that in the top right-hand side of this picture, we have a bacterial pathogen with some PAMPs, or those pathogen-associated molecular patterns. Well, these dendritic cells are going to have PRRs, or pattern recognition receptors, that bind to those PAMPs. That's going to lead to the engulfment of the pathogen, and a phagosome is going to form. That phagosome is going to decrease in pH to begin to destroy the pathogen. The phagosome fuses with a lysosome to release digestive enzymes to further degrade this pathogen. Here's where we also see the oxidative burst, where we might see the production of ROS and RNS, those reactive oxygen species and reactive nitrogen species. And then what's not shown in great detail in this picture here is that after this step, we have vesicles that contain MHC2, which are shown in green, and these vesicles are going to fuse with phagolysosomes. At this point, the epitopes of antigens are said to be loaded onto MHC2, and these antigens then are, with the MHC2 and the vesicles that hold them, are going to move toward the outside of the cell and be exocytosed. And so in the end, what you're going to find is MHC2 on the surface with the antigen loaded in this little binding cleft, as is shown in our image here. And what we say is that the antigens that are being presented, or the epitopes that are being presented, are immunodominant, meaning they trigger a very strong immune response. Well, these are proteins. Proteins are what are going to be loaded into both MHC1 and MHC2. Sugars, DNA, and even in some cases, lipids can stimulate immune responses, but those are very weak antigens. At this point, this professional antigen presenting cell is going to present antigen within the lymph nodes to what we call T helper or TH cells. Now let's talk about presentation on MHC1. All cells that contain a nucleus will present antigen on MHC1. And this even includes those professional antigen presenting cells that we just talked about. In all cells, we find a protein complex known as the proteasome. And this complex helps to degrade proteins once they've been denatured, or oftentimes we think of once those proteins have grown old. But more specifically, we really mean here once they've lost their three-dimensional shape and they no longer can function. Well, the proteasome does this non-specifically. Not only does it break down our own proteins, but also it can break down the proteins from foreign pathogens, for example, viruses and bacteria. And after the proteasome breaks down these proteins, as is shown in this picture, into short peptides that are about 20 or so nucleotides in length, these peptides are transported into the endoplasmic reticulum through a transporter known as TAP, and then these peptides are loaded into MHC1. And so this image here is showing you that these peptides with MHC1 will be exocytosed through the endomembrane system. So they go from the ER to the Golgi to a secretory vessel, and then exocytosis occurs, and that MHC1 is presented on the cell surface. Now, one of the things that's really important to note here is that MHC1 presents both self and non-self epitopes. And so this is really important because we have to train our body not to react to itself. And that is something we're not going to go into great detail with, but is a conversation that we'll briefly address when we talk about T cells. Now, this presentation of MHC1 has also been looked into as a way to train T cells to kill cancerous cells. If, for example, a mutated protein is being presented on the cell surface and it's recognized as non-self, well, then this cell should be forced to die or should be killed by those immune cells that recognize this foreign protein. There are really two main types of cells that do look for the presence of MHC1 and epitopes loaded in MHC1. And these are known as cytotoxic T cells, or TC cells, and natural killer cells, or NK cells. And we'll talk about both of these when we discuss T helper cells. Now again, the role of presentation here is to stimulate and activate the adaptive immune response, and that is that specific response to an infection that occurs maybe days after the initial infection has entered the body. The T cells that are going to be activated in this process do require the antigens to be presented to them in order for those T cells to be activated. And the first site of this presentation is going to occur within the lymph nodes when those professional antigen presenting cells travel to the lymph nodes and present those epitopes to helper T cells. And again, any antigen presenting cell, which is any cell that can place MHC1 on its surface, can present antigens to cytotoxic T cells. After these initial presentations, which occur within the lymph node, those helper T cells and cytotoxic T cells will begin to respond to the site of infection, where they can then continue to be activated or stimulated by further interactions with antigen-presenting cells. And finally, to summarize the difference between MHC1 and MHC2 presentation, recall MHC1 will be found on all cells that contain a nucleus. And so in this respect, any cell can be an antigen-presenting cell. Specifically, MHC1 will be used to present antigens to cytotoxic T cells and natural killer cells. And typically, we oftentimes think that MHC1 is presenting epitopes from inside of a cell. MHC2, on the other hand, is found only expressed from professional antigen presenting cells. It presents to T helper cells, and again, this will be presented on be presenting epitopes from organisms that have been phagocytized.